Good day, students. I'll be your biology teacher for today, and the topic for today's lesson is microorganisms in action. By the end of the lesson, you'll be able to recall what it means to culture microorganisms, name the scientist whose discovery led to a breakthrough in medical science and what it discovered, itemize the various instruments that can be used for culturing microorganisms in the laboratory, state ways of measuring growth in microorganisms, discuss the economic importance of microorganisms, concept of culturing microorganisms. To culture microorganisms is to collect samples of the microorganism from water, air, animals, plants, and various parts of human body, and multiplying the microorganism by letting it reproduce in predetermined culture medium, that is, appropriate food material for scientific study. The principle is similar to growing a crop on a farmland, except in this case, it is microorganisms growing in a petri dish or test tube. This is how scientists grow microorganisms to produce antibiotics. Alexander Fleming was a medical microbiologist. He made one of the greatest accidental discoveries ever, which led to a breakthrough in medical science. He discovered penicillin in the year 1928, the first form of antibiotics, while growing cultures of bacteria on petri dishes in the hospital where he worked. Culture medium can be prepared as 1. Liquid or broth medium, nutrient broth, which can be prepared using 3 grams beef extract, 2 grams yeast extract, and 5 grams peptone. 2. Solid or agar medium, nutrient agar. Agar is a gelatinous material obtained from the marine algae and usually added to nutrient broth, liquid medium, to obtain a solid consistency, which is a good device for the culture of bacteria on a solid surface. Preparation of culture using solid medium. All apparatus to be used must first be sterilized by autoclaving, using pressure pot, or boiling for 15 minutes in a saucepan half filled with water. Measure out your nutrient broth and keep it covered in a sterile flask. Boil the agar to dissolve it and add it to the nutrient broth. Leave to cool. Before the medium solidifies, which it does at about 45 degrees centigrade, pour it into the petri dishes or test tubes you are going to use. Petri dishes should be filled to about halfway and the lid replaced immediately. Test tubes should be kept at a slant while the medium is solidifying. This provides a larger surface area for bacterial colonies to grow on. The microorganism to be cultured is then introduced onto the agar medium. Inoculation. Cover immediately. Clearly label the petri dishes and test tubes and place in warm but dark compartment or incubator. Incubation. Petri dishes are best placed upside down to reduce the chances of contamination. Observe and take records for 2 to 3 days. Precautions to be taken during preparation of culture. Wash your hands with soap before and after the preparation of agar solution. All instruments should be sterilized before the beginning of culture preparation. Open petri dish only slightly and cover at once. Close petri dish firmly with adhesive tape. Sick person should not be permitted to take part in culturing experiments. Avoid talking, coughing, sneezing, and touching of infected jelly. We'll be back after the short break.